Hello, good morning. Time for next past paper. Well, here we go. Unit 1, Paper 2, June 2018. Question 1A. In the space below, draw and label the chemical structure of a typical phospholipid molecule, clearly indicating its three major components. Label the hydrophilic and hydrophobic parts of the molecule. Alright, so this is what your drawing will look like. And remember to say the chemical structure, so you're not gonna draw just a little circle with two tails coming out, right? That would only probably give you one mark. So what you need to do here is you need to put all the molecular structures here. So here we have the phosphate head, we have the fatty acid tails, right? And remember we have two, and we have the glycerol molecule bridging the two of them. The hydrophilic region is the phosphate head, the hydrophobic region are the fatty acid tails. Now remember what hydrophilic means. It means having affinity to water, water loving. Hydrophobic means water repelling or water hating. So the fatty acid tails are usually facing away from the cell cytosol and the exterior of the cell. They're sort of bunched up in the middle of the phospholipid bilayer. Once you have the three parts here labeled and drawn, you should be able to get your 4 marks. B. Describe how phospholipids are oriented to form the lipid bilayer of cellular membranes. Alright, so remember I said what will happen here is that the phosphate heads are going to be facing outward and the hydrophobic tails, which are the fatty acid tails, are going to be facing inward. So the phospholipids form the bilayer by having the hydrophilic heads facing outward, the tails facing inward, and those tails there are going to be sandwiched between the bilayer, away from the cell exterior and away from the cell cytosol, because those they are water based, and there's going to be 2 layers of phospholipids that form the bilayer. Pretty simple to get to 3 marks. C. State the name of the lipid responsible for maintaining the fluidity of cellular membranes. That lipid is called cholesterol. So what the cholesterol does is that it maintains the fluidity but also the rigidity of the bilayer. Let's say for example that in a very cold temperature and those phospholipids tend to bunch up together, right? They tend to get too close. Where we have a problem there, we wouldn't have much not facilitated with it, we wouldn't have much simple diffusion occurring where oxygen molecules, for example, could pass through the bilayer. So what cholesterol would do is ensure that there's enough spacing between those phospholipids. D. Describe how intrinsic proteins allow the movement of substances across cellular membranes. Here we have a phospholipid bilayer, and we have 2 types of proteins that would be here. We have extrinsic proteins, and those would be on the periphery, on the outside. They basically would not span the entire bilayer, and typically you would have proteins or lipids attached to this called glycoproteins or glycolipids. However, intrinsic proteins, those are the proteins that span the entire width of the bilayer. And what could happen here is that these could be used as channels or they could be used as carriers. So a channel, for example, would be able to move substances in and out. You also have ones called aquaporin, which are specific to water. They could also work as carriers, which means that they're kind of like gated channels. And for example, you might have one that needs to attach a specific molecule onto it before substances can pass. An example of this would be sodium potassium pumps that would be in the neurons. So looking at the question here, intrinsic or integral proteins span the entire length of the phospholipid bilayer. They can take the forms of channels which allow the passage of large polar materials. An example of that would be glucose or mRNA in and out of the cell via facilitated diffusion. They can also form carrier proteins, which are gated channels activated by certain factors such as voltage or chemical or molecule called a ligand. These usually require ATP to activate, and thus this is called active transport. Once you have 2 points well laid out, you should be able to get your 4 marks. Part E. Figure 1 shows the molecular structure of a cellular lipid, and here we could see that it has 1, 2, 3 fatty acid tails. That means that it is tri, and because it is a lipid, that is a triglyceride. 2. Explain how two features of the biomolecule in figure 1 are related to its function in animal tissues. Well, triglycerides, or which is a component of fats, they contain a high amount of energy. And in fact, you have 3 fatty acid tails, and those store more energy due to the long carbon chains with hydrogen bonds. So you'll see here that, remember, carbon would have 4 things bonded to it, or would have 4 bonds. So each carbon here, aside from the last one, would have 2 hydrogens bonded to it. When these hydrogen bonds break, that releases energy. Now, because these chains are so long, there's a lot of energy that can be released by the lysis of triglycerides. Lipid properties allow insulation or protection as adipose or fatty tissue builds up with animals, and triglycerides are needed to absorb the vitamins A, D, E, and K. Vitamins B and C are water soluble, not fat soluble. Let's look at 2A. Seed types in pea plants is determined by a single gene with round seed, capital R, dominant to the wrinkle seed, common R. 3 pairs of homologous chromosomes, homologous means that the pairs have the same shape, are depicted in figure 2 with the gene locus. The locus means the position that the gene is held in on the chromosome for seed type indicated by X. We have three pairs here, 3 pairs of chromosomes A, B, and C. And they said in figure 2, insert the appropriate alleles which correspond to the phenotypes A, B, and C given the following characteristics. So A is homozygous round seeded variety. So remember, round is dominant, so you're going to have a capital R on one locus and another capital R on another locus. B is going to be a homozygous wrinkle seed variety. So homozygous, remember, means homo means same, so 2 of the same. So this is going to be a recessive R on one locus and a recessive R on the other. Heterozygous variety now means that the hetero means different, which means you have 2 different alleles. So on one locus you're going to have the capital R, the dominant, and the other locus you're going to have the common R, the recessive. Alright, let's look at part 2. Draw a Punnett square in the following box to illustrate a test cross involving a pea variety which is heterozygous for seed type. State the ratios of the phenotypes for the offspring. Okay, so in this case here, we're going to make this parent heterozygous, which means they're going to have a dominant and a recessive. And we're going to make this one homozygous recessive. So they say it has to involve at least one heterozygous, which is this. So this first possibilities here is going to inherit this dominant and this recessive here. The second possibility is going to inherit the dominant R and the recessive R. This third possibility is going to inherit this recessive R and this recessive R. So this is going to be a wrinkle seed. And the last one is going to inherit this recessive R and this recessive R. And this is going to be a wrinkle seed. And the genetic ratio, the genotypic ratio, is going to be, so there's 2 heterozygous and 2 homozygous recessive. So it's going to be 1 capital R to common R to 1 common R to common R. And the phenotypic ratio is going to be 1 round to 1 wrinkled. Distinguish between a gene and allele. A gene is a sequence of DNA nucleotides that codes for a particular polypeptide or protein. What would happen here is that the DNA, well, that gene sequence is going to have an mRNA equivalent, and that is going to be read by a ribosome and translated into a sequence of amino acids, and eventually that will fall into a protein that would then express a certain trait, whether that be blue eyes or round seeds or dark skin. Now, an allele is one or more alternative forms of a gene. So using the previous example with the seeds, the letter R expressed whether the seeds would be round or wrinkled. But capital R and common R are alleles for that trait, where capital R means round seeds and that is dominant to the wrinkle seeds, which is common R. Those are different forms of the gene. Let's look at B. A micrograph of pressed onion meristem cells is reproduced in figure 3 with a scale bar of 10 micrometers given in the lower left corner. Here you can see that. And there are 3 cells labeled at different stages of mitosis. So there's 3 cells labeled here. There's D, where inside of the cell we could see there's more chromosomal material. The chromosomal material is a little more apparent. E, where we can see it looks like the chromosomes are moving apart from each other inside the cell. And F, we could see that, well, this cell is a little more vague with its genetic material. So we'd have to label these 3 here. Now D is going to be prophase. And this is where the chromosomal material is a little more condensed, is a little more visible. E is going to be anaphase. And this is where the chromosomes are going to start moving to the poles of the cell. They're going to get pulled apart by spindle fibers away from the what you call the equator of the cell, and they're going to go more to the poles. And this would be right before telophase, where 2 new nuclei would be started to form before cytokinesis, basically the splitting of one cell into 2. And this last phase here is interphase, which is not really a phase of mitosis. Interphase is the beginning of cell division. It actually accounts for 95% of the cell division process, because what's really happening here at interphase, where the nucleus and the nuclear membrane is still intact, basically, is that the DNA is being replicated inside here to form that double chromatid structure that we see as chromosomes. And here we have the names of the 3. D is prophase, E is anaphase, F is interphase. 3A. Dental amalgam fillings are composed of approximately 50% metallic mercury. Mercury is a toxin and has been linked to neurodevelopment disorders in babies. Table 1 shows the relationship between mercury fillings and the concentration of mercury in the blood. Let's look on the table here. So the table shows the number of maternal amalgam fillings, and you'll see we have 2, 4, and 9. And then we have the mercury concentration in microgram per liter. Here we have in the maternal blood. Here we have in the fetal blood. What can we observe just by looking at the table? Well, we can observe that the higher the number of fillings is basically the higher the amount of mercury concentration in both the maternal blood and the fetal blood. So this is what a graph would look like. You want to draw a bar graph, and you want to make sure that you have the separations between the two. You also want to make sure that you have a key or legend to represent fetal and maternal. Here we have the number of maternal amalgam fillings, and here we have the mercury concentration in the blood. Make sure and put the units, which is microgram per liter. You want to make sure that your graph occupies about half of the space, at least half of the space on the grid, on each axis. And what can we observe here? We can see that the fetal mercury concentration is always higher than the maternal. But with each one, whether it's maternal or fetal, there's an increase in mercury concentration when there's an increase in maternal amalgam fillings. So part 2, state 2 conclusions based on the comparisons shown on the graph, basically what we just mentioned. The concentration of mercury in fetal blood is higher than the maternal blood, or vice versa, maternal blood is lower than fetal blood. And 2, as the number of maternal amalgam fillings increase, the concentration of mercury increases in both maternal and fetal blood. The 2 points here simply explain what give you 2 marks. Explain whether the movement of mercury from mother to fetus is an active or passive process. Now this would be an active process, and let me explain why. Remember that the maternal mercury concentration was always lower than the fetal. So that means that for it to go against a concentration gradient, basically from a lower to a higher concentration, this would need ATP to do so. Why? Because the fetal blood always has a higher concentration than that of the mother, so the movement must occur against a concentration gradient. B. Figure 4 is a cross-section of a mature pollen grain. Write the correct labels for A, B, C, and D. Well, A is called the intine, and that would be the inner layer of the grain itself. B is going to be the X sign, and that is going to be the waterproof outer layer. C is going to be the tube nucleus, and how you'll differentiate that from the generative nucleus, which is more elongated. The tube nucleus is more round. Now the tube nucleus is going to form the pollen tube that will grow down the style to deposit the male nuclei. The generative nucleus is going to divide into two male nuclei. One is going to fertilize the egg. One is going to make the endosperm nucleus that forms the endosperm nutrient source. C part 1, describe the events following pollination which lead to double fertilization. Well, first the pollen grain is going to germinate to form the pollen tube. This usually happens when the pollen grain attaches to the stigma, probably takes in some water and sucrose, and then the grain ruptures to let forth the pollen tube. The pollen tube grows down the stigma. So on descent, the generative nucleus divides by mitosis, and that produces two male haploid gametes. Once in the embryo sac of the ovule, one male haploid gamete fuses with the ovum nucleus, basically the egg cell, to form the diploid zygote, and the other haploid male nucleus fuses with the diploid endosperm nucleus in the embryo sac to form a triploid endosperm. The endosperm, you can think of that as the placenta, sort of like a placenta for the developing embryo that provides food and nutrients to sustain its growth and development. So C part 2, state the significance of double fertilization to the human diet. The term double fertilization, what that means is what I previously described, where one male nucleus fertilizes the egg and the other male nucleus forms the endosperm. The endosperm nucleus. The endosperm, as I said, is the food source. Now in some plants there, the endosperm actually remains in the food, and that endosperm actually provides the main source of nutrition for human consumption. Examples of this would be in rice, wheat, and maize. Now, after fertilization, the ovary usually becomes a fruit, and the ovules become seeds. The ovary wall becomes something called the pericarp. And the pericarp is what contains a lot of sugars. For example, if you bite into a mango, you are eating the pericarp of what was once the ovary wall, and that there contains sugars. So sugars are contained in the pericarp, which was once the ovary wall. 4a. With reference to the structure of enzymes, distinguish between competitive inhibition and non-competitive inhibition with respect to enzyme activity. Well, first, let's understand what is an enzyme. Enzymes are globular proteins with a three-dimensional shape and tertiary protein structure. Enzymes, their mode of action is usually they would attach a substrate, which is a complementary molecule that fits into its active site. The active site, think of it as a cleft or depression on the structure of the molecule. So this specific substrate would have, let me say for example, a specific set of amino acid sequences that would fit or combine with or bond with the amino sequences on the active site. That would allow a reaction to occur and would either allow the substrate to undergo lysis, which means that it would break down, or it would combine or bond two substrates together. So how competitive inhibition occurs is usually what would happen here is that this substrate is supposed to fit into this active site. And this would basically initiate the reaction to turn the substrate into products. However, what is happening here is that there's another molecule called an inhibitor or competitive inhibitor that is, think of it as it is blocking the active site, or is occupying some space in the active site. Now, the thing about the active site is that it has a high specificity, which means that the substrate has to have a specific shape in order to fit into this little depression here. So if something else is blocking it, or something else has this specific set of amino acid sequences, at least to bond to part of the active site, this would prevent the substrate from attaching. So this is what is called competitive inhibition. Non-competitive inhibition, you'll notice here, is where the active site, if we look at the shape of it here, and we look at the shape of it here, we can see that the active site has changed a little bit on the one on the right. Well, why is that? Well, what has happened here is, you notice that nothing is really attached to the active site. But what has happened here is that we have a non-competitive inhibitor attached. And this could attach to another depression or another sequence of amino acids on the enzyme. So this part here, where something else could attach, and nothing is really supposed to attach here. Right, so this could be something that is lethal. It could be a kind of poison, for example. So this here, this is not called an active site. This is actually called an allosteric site. And the word allosteric, the prefix allo means other. So basically, this means like another site that things could attach to, but not really supposed to. So what happens here is that when substances attached to the allosteric site, the whole enzyme sort of changes shape a little bit. So imagine like it gets warped. So the non-competitive inhibitor, which is not competing for the active site, changes the shape of the enzyme so the active substrate now cannot fit into the active site. This could be quite dangerous, especially if the enzyme is permanently altered or the non-competitive inhibitor permanently bonds to the enzyme. So to explain here, competitive inhibitors have a similar shape to the substrate molecule. They fit temporarily onto the active site and compete with the substrate for entry to the active site. So the effect of competitive inhibitors decrease if you have more and more substrates, which give the substrates more and more likelihood that they would bond to the enzymes or go into the active site rather than the inhibitors themselves. Non-competitive inhibitors bind to another part of the enzyme molecule called an allosteric site, or it may bind permanently to the active site. So it may get stuck there and the active substrate cannot actually bond with the enzyme to form the complex to break down into products. What this does is that it distorts the three-dimensional structure of the enzyme. Distortion of the enzyme structure can prevent the substrate from binding, and the effects of non-competitive inhibitors are not reversed if substrate concentration is increased. The reason being is that these can potentially permanently alter the enzyme, and they attach to different parts of the enzyme, right? So they don't really compete for the same space. Alright, let's look at B. Discuss the role of 4 tissues found in the roots of a plant that support the major functions of the root. So any 4 of what I have here will work. We have the epidermis, the parenchyma, the endodermis, the xylem tissue, the phloem tissue. You can also use pericycle or any meristematic tissue on the roots. What you just have to do is put a basic description of their role. So the epidermis is the outermost layer of the root, and that can offer some protection as a physical barrier. Now remember, they also have root hair extensions which aid in osmosis and provide a large surface area for the uptake of water and nutrients. The parenchyma is located beneath the epidermis or the cortex. These cells are relatively unspecialized. So what they basically do is that they facilitate the movement of water from the root hairs towards the center of the root where the vascular bundle is. So they have thin cell walls and they provide support when cells are turgid. Some of them might even have amyloplasts or starch reserves. The endodermis, which is something that you'll learn a little more about in Unit 2, but this is a single layer of cells surrounding the stele or the vascular tissue. And typically what you'd have is some of these cell walls are going to be waterproof to form something called a Casparian strip. The Casparian strip, what that does is that actually regulates the movement of water and mineral salts into the vascular tissue. A lot of it is impermeable, is actually made up of a material called suberin, which is actually the same material that they use in wine corks. And what that does is, I say, it acts as a waterproof barrier. Next, we have the xylem tissue, any vascular tissue. Of course, this contains dead, empty cells, which are long, hollow, and continuous with lignified cell walls. Lignin is a dead material that's used for support. There's no end wall, so it's continuous. Lignin gives strength to the organ for anchorage. It keeps the plant in the roots, keeps it from toppling over. Vessel elements are arranged end to end to form continuous tube for water transport throughout the plant. Now we have the phloem tissue, which contains phloem sieve elements. And phloem tissue, unlike the xylem, is living. So these are living cells with perforated end walls called sieve plates, arranged end to end. So they are consisted of little elements. It's not long and continuous. What this does is that it allows transportation of sucrose, which is called translocation, to flow to the root and other parts of the plant to supply energy and building materials. So what the phloem does is that it transports sugars from photosynthetic organs such as leaves to non-photosynthetic organs such as roots that can't make, their own glucose and their own starch as a result. 5a, with the aid of a diagram, explain how the information stored in a gene is used to synthesize RNA. So let's take a look at this diagram here. What is happening on this diagram is typically what we'll have is the unwinding of DNA. Remember, the DNA is a helix consisting of two strands. There's a strand that runs from the direction 5' to 3', and one that runs from 3' to 5'. On each of these strands, there are nucleotides, and at the end of each nucleotide, there's a base, a nitrogenous base, which could be thymine, adenine, cytosine, or guanine. Each base, of course, pairs with, it has a complementary pair. So I like to think of it as car in the garage, which is C and G, so cytosine pairs with guanine, or apple in the tree, which means that adenine pairs with thymine. Now remember, that's with DNA, but RNA doesn't have thymine, RNA has uracil. So you could think of it as apple under the tree. Apple in the tree for DNA, apple under the tree for RNA. A pairs with U. What's not depicted on this diagram is an enzyme that is going to unzip this helix, right? And the enzyme that does this is called DNA helicase. Helicase means it's an enzyme that basically unravels the DNA helix. It's a helicase. And this is going to start unzipping the DNA. And what you'll notice here on this diagram is that a piece of mRNA is being built from one of these strands here. So the piece of RNA is actually being built from the 3' to 5' strand, right? And what is happening is any free RNA nucleotides that is in the nucleus at this point, that is going to be attaching to the DNA strand. So for example, if the DNA strand is TACC, the nucleotide for A is going to bind with T, nucleotide for uracil is going to bind with A, nucleotide for guanine is going to bind with C, and so on. And this is going to create an elongated mRNA molecule. Now, what facilitates all of this is another enzyme called RNA polymerase. If we break this down, well, RNA is a polymer. Polymer, remember, is just a sequence of monomers, right? A sequence of nucleotides, and a polymerase is an enzyme that builds a polymer. So our RNA polymerase is reconstruction worker here that is helping to build this thing. While DNA helicase is like the thing that's going to unzip the DNA to allow this to happen. It's not that difficult. So first we're going to explain what is a gene and what is mRNA. The information stored in the gene is in the form of a sequence of bases, which is used to sequentially arrange amino acids that would eventually be translated to form a protein. So mRNA is the molecule which is used as a template to organize the primary structure of proteins, basically the polypeptide chains that are being built by the ribosomes. The process by which DNA is used to synthesize RNA is called transcription. So that building, that elongation of the RNA molecule that I would have just shown you, that building of it with the RNA polymerase, that is called transcription. So that produces mRNA with complementary base sequences to one strand of DNA. The DNA would have unzipped and both strands would have separated, and this would have been catalyzed by DNA helicase, the DNA helix enzyme. One strand is being used as a template strand, so the mRNA is being built off of one strand of DNA, and complementary bases are added to the strand, and this is copied in a 3 to 5 prime direction, catalyzed by RNA polymerase. This is just the enzyme that builds the RNA polymer. A pairs with U, apple under the tree, and C pairs with G, car in the garage. Condensation reactions occur, which allow phosphodiester bonds to form between the bases to form the mRNA, and this is formed in the 5 prime to 3 prime direction. 3 bases or triplet codon on the strand, so for example AUG, that would code for a particular amino acid. So this is a whopping 10 marks, so you want to make sure and have as much detail as you could to describe this process of transcription. Let's look at B. Using an example, briefly explain how proteins are responsible for the phenotype of organisms. This is a 5-mark question. So this one is basically what you want to do is you want to start from proteins and you want to explain how proteins express traits, right, in cells and in an organism. So the phenotype, first of all, is any external observable characteristics of an organism, and this is determined by the kinds of proteins produced in the body. A very good example of this is melanin. The more melanin proteins that someone has in their basal epidermal layers means that they appear darker skinned in color. The less melanin that they have in the basal epidermis means that they are lighter skin in color. This is a basic example of how a protein can influence appearance or phenotype in this case. So proteins are expressions of genes. A particular gene codes for a particular protein. You'd have a particular gene that would code for collagen, one that would code for keratin, one that would code for particular enzymes like amylase and pepsin and renin. Cells of a similar phenotype would form tissues. And of course, different tissues form organs. Organs form organ systems. And together, that forms an organism. So this is how proteins, for example, would be expressed in an organism, because those proteins will be found in cells. Those cells make tissues. Tissues make organs. For example, organ would be the skin in terms of melanin, and those organs come together to form an organism. So 2 examples I give here. The protein hemoglobin is packed into the erythrocyte as a red blood cell, given its characteristic red color and biconcave shape. With melanin, when there are larger amounts in the basal epidermal layer of the skin, it would appear darker in color. Melanin is the protein. The dark skin is the phenotype. Let's look at question 6a. Outline the steps involved in plant tissue culture and explain the key scientific principle underlying this technique. Alright, so here we have 7 points because this is 7 marks. So first thing that's going to happen is that a small group of cells from the plant is going to get removed, and this is called an explant. And why just a small group of cells? This is because these cells, each cell actually contains all of the information required to produce the entire plant. These are called totipotent cells. And basically what totipotency means, it comes from the Latin totus, or kind of like total, which means whole. And of course potent, which means strong. So it means that each cell actually is strong enough to develop into an entire plant, which is kind of amazing. So after that small group of cells are removed from the plant, that explant, is gonna have to be disinfected, because it's just a small group of cells that is very susceptible to harmful fungi and any other pathogens. So it's gonna be disinfected, and the explant is going to be immersed in a sterile aerated solution or culture medium containing hormones and nutrients. So these growth hormones that usually be there, this would expedite cell division, because a few cells could get wiped out very easily by some pathogens. That's why it had to be in a sterile environment. So the undifferentiated cells of the explant divide repeatedly to form a callus. And this callus can be maintained indefinitely in the culture medium. These callus cells are then subcultured on a sterile medium and induced to form shoots and roots by varying plant growth substances. So the nutrients that this is going to be fed with is going to be like nitrates, some sucrose. There's going to be some hormones like auxins in there that's going to stimulate growth, right? The plantlets, when they grow, so when they get a little bigger, they're going to be transplanted to sterile soil once they are large enough. Still, they are in a very susceptible stage, so they have to usually be kept in a laboratory, in a very sterile greenhouse kind of structure before they can be transplanted somewhere else. And this is the last question here. B. Complete table 2 by comparing the structure and function of the following 4 features in a human sperm cell with those of a secondary oocyte, which is the female egg cell. The first one here, the overall structure and size. So the sperm cell is differentiated into a head, which contains the acrosome and the nucleus, the neck, the middle piece, which is the body that contains mitochondria, and the flagellum or the tail, which allows motility or allows it to swim. The oocyte, on the other hand, is not really differentiated into different parts like that. It is multi-layered. So it's a spherical cell that's surrounded by follicular cells and the zona pellucida. And in terms of size, it is actually much larger than the sperm cell. It contains more cytoplasm and has to store more nutrients. The nucleus of the sperm cell is haploid, and this delivers the male chromosomes. The sex chromosomes for a male is going to be either going to be an X chromosome or a Y chromosome. For the female is also haploid, and this delivers female chromosomes. The female sex chromosomes are just going to be X, no Y. The cell membrane of the sperm cell contains glycoproteins that facilitate the union with the oocyte. So the cell membrane is going to also have receptors that think of it as going to activate during capacitation when they're swimming towards the secondary oocyte. But basically what would happen here is that the sperm cell would be able to bind to the oocyte in order to release its acrosome so it can send the nucleus into the cell so it can form a zygote. The egg cell, on the other hand, the oocyte, contains microvilli that absorbs nutrients. It has proteins that bind to proteins on sperm cells that facilitate entry of the sperm cell nuclei. Finally, in terms of mitochondria, the sperm cell and the oocyte both have numerous mitochondria. Whereas the mitochondria for the sperm cell provides energy for motility or swimming, the mitochondria in the oocyte is more used for providing energy for metabolic processes and development, because that egg cell there has to be sustained for a few days while it is going through the fallopian tube, and it is undergoing periods of development if it does get fertilized to form a zygote, because it has to undergo mitosis several times to become a blastocyst before it gets implanted into the endometrium or the uterus lining. Alright, so that was June 2018. Take care all ya. Laters.